Hello and welcome back from our lunch break. I'm Matthew Savari from UCLA's TDG. As we all get settled in, I'd like to take an opportunity to remind everyone to make sure you're using your Jujama app to get most out of your conference connections. That said, I'd like to hand the stage over to Dr. Thomas Carmichael, Acting Director of the Broad Stem Cell Research Center, who will be moderating our next session, CIRM Strategic Opportunities and Beyond 2020. Dr. Carmichael.
Good afternoon. Thank you and welcome to the first afternoon of LA Best. It's a pleasure to be a moderator of this symposium, which will be a really exciting, diverse, and dynamic talk among some of the experts in the field around the topic of CIRM strategic priorities and opportunities beyond 2020. We have Dr. Andy McMahon, Dr. Maria Millan, Mr. Al Rowlett, and Dr. John Zaia. And we'll take a moment for each to quickly introduce themselves and then go through the panel. I'll set the stage after those introductions. So we'll start alphabetically. Dr. McMahon.
Hello, I'm Andy McMahon. I'm the director of the Eli and Edythe Broad CIRM Center for Regenerative Medicine and Stem Cell Research at USC.
Thank you. Dr. Millan.
Good afternoon. I'm Maria Millan, the president and CEO of CIRM. Great to be with everybody today.
Great, thank you. And Mr. Al Rowlett.
Good afternoon. I'm Al Rowlett, a member of the ICOC board and the Behavioral Health, Mental Health Patient Advocate.
Great, thank you. And Dr. John Zaia.
Hello, I'm John Zaia. I'm the director of the Center for Gene Therapy at the City of Hope. I'm a clinical trialist and I do a number of gene therapy trials. I'm also director of the Alpha Stem Cell Clinic at City of Hope.
Great, thank you. As you can see from the constitution of the panel, we have fairly distinctive elements represented. And as we look back, if the panel is looking at CIRM beyond 2020, and we look back at the CIRM in the prior epoch, we see a lot of novel things they did with the Alpha Stem Cell clinics, with the Bridges and Sparks training programs, the CAPS or the clinical advisory panels, the Cell and Gene Therapy Center. There were really a whole network of very interesting things that CIRM developed. And it will be a part of this panel to kind of explore what the future holds. And a good way to start that off was with Dr. Millan as director of CIRM, to kind of hear her perspective on CIRM strategic priorities in the coming CIRM 2.0. So Dr. Millan.
Thank you so much, Dr. Carmichael. Before I go on to what we hold in the future, I think it's really important to talk about where we are today and how we got here, if that's okay. So CIRM story actually began in 2004 with a $3 billion bond initiative under Prop 71. And we call this the Prop 71 era, which thankfully just ended in November this past year when Prop 14 provided for a reauthorization of $5.5 billion in funding to support stem cell regenerative medicine research and to continue to build this capacity in this ecosystem in California. So I also want to put in context, when the agency was first formed in 2004, this really wasn't even a field as we know it today, and actually not really a field where investigators and scientists could gain meaningful support for the research. I mean, if we think about it, it was only 6 years prior to that the human embryonic stem cell was isolated, and it was literally the year before, in 2003, when the human genome was sequenced under the International Human Genome Project. So quite an amazing history where CIRM started. Fast forward to today, CIRM's funded over 1,030 programs with 70 clinical trials, predominantly first in human clinical trials using novel approaches with cell and gene therapy. So that's quite remarkable. Again, in looking to the future, we want to talk about what kind of the value creation and value proposition of CIRM was that's been created in this Prop 71 era. And I would say that it's noted both for an acceleration funding model that's predictable, that goes with it, as Dr. Carmichael had mentioned, the support regarding advisory panels and infrastructure to support this novel field in a regulatory manufacturing arena. And also creation of first-in-kind type infrastructure such as stem cell, the largest stem cell repository for research to induce pluripotent stem cell bank, and a stem cell genomics hub. Importantly, the patients are kept in the center, and the creation of the future workforce, as you mentioned, Dr. Carmichael, with the Bridges program, et cetera, is a priority with thousands of students being trained in the field and going on to be leaders themselves. I think that I'd like to then pause there and just to say that we have a wonderful starting point to build upon, and we have quite a future, but I'm going to pause right there.
Okay, really great, interesting description. We have a structure here where we're going to have about 10, 15 minutes from each panelist and then take questions at the end, which we decided was probably the best for flow. Dr. Millan, you have a little bit of time to discuss what you see as the priorities and the projection into the future. What's CIRM Prop 14 2.0 looking like?
So by statute, the Prop 14 had provided for an earmark in funding for diseases of the brain, for research in diseases of the brain and neuropsychiatric diseases, which is new. It also opened it up so gene therapy and genetic research are accounted for, whereas CIRM had already funded this type of research under Prop 71 as vital research opportunities. Now under Prop 14, it was anticipated and provided for along with funding stem cell research. In addition, the proposition also provides for creation of what's called shared labs. We're envisioning these as competency cores and resources that could enable the entire scientific community and their partners, as well as this idea of accessibility and affordability, which is around the idea of we can take this amazing science, translate it, de-risk it, get investment, but how do you truly get it out to those in need? And those are some of the topics I'm really looking forward, because there are many, the other panelists are very involved in other aspects of this, including our board member, Al Rowlett, who has been a leader in helping us think through this. In addition, we will continue, obviously, to fund world-class science, basic science, translational science, clinical research. So how we envision kind of the next generation in funding is the opportunity to organize the way we do things in a way that really leverages the successes and also the learnings from each of the individual projects through a team science and consortium type models. And this will enable both the efficient use of some of the shared labs and competency hubs that I mentioned, as well as building a knowledge network. So for real data sharing and leveraging that to use in a responsible way to advance the research and highlight the key needs. I want to highlight basic research, which I'm sure Dr. McMahon will have his own points on that. But when we think about translational research and getting to the clinics, none of that is possible without strong science. So the pipeline absolutely 100% relies on continuing to support discoveries and to support basic mechanistic research, and then also supporting reverse translation, where we learn things from later stage programs and bring it back and study it again in the lab, whether at the submicroscopic level or the cellular level. So I hope that kind of frames it. And so the process we're going through is to actually put the tactics behind this and test assumptions and determine feasibility. And then, as you know, how CIRM works is that those get brought forward after the strategic plan is brought to the board for final approval. We start rolling out the concepts that then lead to program announcements. And that's how CIRM as a funding agency is able to make these important ideas into reality. So I can continue to take questions or elaborate on this more in a little while.
Great, thank you. And you can submit questions, I'm reminded from an organizer through the chat. You can submit questions through the Q&A and we'll address them right after we're done. So the flow that we thought would work well here is if we talk about some of the basic science and overarching scientific ideas with Dr. McMahon next. Dr. Zaia will talk a lot about building the translational pipeline and what that looks like and applying to patients. And Mr. Rowlett will then talk about how we engage diverse communities, we deal with the social determinants of health, and we start to think about stem cell biology, CIRM, and mental health. And so Dr. McMahon, you're next. Thank you. You're on mute, Andy.
Wonderful to have this opportunity to speak today. Much of CIRM in the public's mind, I think, is framed around stem cells. And stem cells are acting critically in our bodies every day to renew various cell types in different organ systems. And when we're injured, stem cells kick in a big way in many of those systems to repair and regenerate. As you know, if you just listened to the previous talk by Don Cohen, there's been great strides over many decades in using stem cells as treatments, particularly for hematopoietic stem cells. So the hematopoietic stem cells can directly regenerate tissue, and that's behind curing cancers, for example, by a bone marrow transplant. And stem cells can be vehicles to curing disease. So you can introduce genes, but gene modification into those hematopoietic stem cells to cure genetic diseases. Stem cells have been really advanced and pioneered through the hematopoietic system. Many of our adult tissues have stem cells underpinning them. And clearly there's a great opportunity to working with adult stem cells in the future to help in repair and regeneration and diseases of those organ systems. Not all organ systems, however, have stem cells. So, what happens there? I'll refer a little bit later to the heart and the kidney, for which there's no strong evidence of large roles of stem cells within those systems. So there we have pluripotent stem cells that have the ability under the right conditions really that emulate how the embryo develops into different organs in the uterine environment, to be able to take those pluripotent stem cells and generate any cell type of the body system. Again, this is really in its infancy. And I would imagine that the use of pluripotent stem cells and the forming of therapeutic cell types is going to be a key part of work that will go on in the second CIRM. But I think that we have to also think beyond stem cells to meet the goals of regenerative medicine. That is what CIRM is. It's regenerative medicine is what CIRM's about. I mentioned that the heart and the kidney have minor or no stem cell roles. And so what about these systems? So the kidney, which is a system I happen to work on, it does have some repair capability, but it's a different type of capability from stem cells. It doesn't have these special cells set aside like stem cells that have the ability to regenerate themselves and then differentiate into the multitude of different cell types in the organ. But what it does is it takes the differentiated, specialized cells and it de-differentiates those on injury and then kicks them into a repair process. So a very different type of a system, but it's not as efficient a system as stem cells. Can we think about ways in which we can augment and improve those normal repair processes? In the heart, it doesn't even have that. But in the heart, people have shown that it's potentially possible to reprogram the cells that are left scarring the heart after a heart attack and actually switch them back to cardiac myocytes and restore injury through an in vivo reprogramming process. At least this has been shown in animal models. Many of those organs that also get injured have very common pathology signatures. So those injured tissues excite the immune system, they generate inflammation, and that inflammation has long-term effects. In fact, there's very good evidence that that leads to acute to chronic disease in the kidney and the loss of kidney functions. Should we be targeting that common pathology that those organ systems have to actually prevent the degeneration of organ systems as a tactic in regenerative medicine? Basic research, we mentioned the importance of basic research for a greater understanding. You know, we have to be a little bit humble here. If we look at our friend, the salamander, if you cut off the salamander's limb, it can regenerate an entire limb. So it makes new muscle, it makes new bone, it makes new tendon, it makes new cartilage, it makes new skin. And it does that in a way that entirely mirrors the normal structures of the limb that was amputated. So I would just end by saying that there is a lot to be learned from basic research about things that are very exciting, that we don't know how they work. And if we did know how they worked, then there's great potential for advancing regenerative medicine in the bigger picture. And I would just make an observation that every 5 years, something comes along that's an accelerator for science. You can just go back every 5 years and find that. The latest accelerator for science has been single cell technologies. Before that, it was CRISPR editing. I can't predict what the next accelerator is going to be, but we do need agencies like CIRM that are actually enabling those accelerator generators to find those discoveries.
Great, thank you. Really interesting. Your thematic summary, Beyond Stem Cells to Meet the Goals of Regenerative Medicine, might be a future seminar. Well said, thank you. Dr. Zaia, you've done a lot of work translating specifically cell and gene therapies, and have a really interesting perspective from that expertise, and also, as we discussed in our panel prep, on how we might move that into particularly difficult to reach populations.
Thank you very much for inviting me. I just want to begin by reiterating what Andrew said, and that is that in order to bring new therapies to the clinic, we have to have methods that work, and those methods derive from science. And it is CIRM's support of science, I think, continued support, that gives us this predictive and recurring ability to imagine that it's possible to make new discoveries that will bring cures where they weren't cures before. That's just the obvious. But in this area in particular, we want things to happen fast. It's not easy to happen fast because it's very complex. The biological systems, again, that Andrew outlined are so complex and so different. So the metaphor I like to use is the one of the moonshot. That is, we made it to the moon, but Goddard first launched his liquid fuel rocket in 1926. And so it took a good 40 plus years for us to get to the moon. And other people said, well, I don't want to just go to the moon, I want to go to Mars. Well, it's taken us longer than to get to Mars. And the same thing is true here. Some people want to cure sickle cell disease, let's say, and they did it in a very excellent way, as you heard from Don Cohn. Other people want to modify the brain. We want to, prevent or repair the brain when we see Parkinson's, we see Alzheimer's. I think that's a much more difficult area to go to. How did Don Cohn make it work and others? He basically took that rocket that he was given, let's say the lentivirus, and he took it apart and reconstructed it and made it more efficient so that he would have the thrust, let's say, to get to a cure. So I don't want to beat this metaphor to death, But that's the idea. What CIRM is providing is a milieu in which this can occur. And they're leading us down this road, and there's going to be bumps along the way, but I think that this is where we want to go to eventually have the ability to cure diseases that currently are not treatable. Now, there's elements of this that are crucial. That is, how do we learn how to do some of these things? And that's where CIRM came up with the idea of an alpha stem cell clinic. And as you know, there are 5 sites, really 6 if you can consider UCLA and UCI in California now. And what they have done for the last 5 years is really set up a system that allows an efficient acceleration of a very difficult clinical trial protocol. So whether it is based on autologous transplant of genetically modified cells, or if it's an injection of let's say, embryonic cells into the spinal cord, all those things had to be learned, as you need a nursing system to oversee it. You need to know how to credential those nurses. You need to then establish best practices. And that's one of the things I think that has been done well by the alpha stem cell clinics. They also form a network that allows them to help each other and even interact with industry. So if industry has a new trial, they would like to see where it could be put into place, where there's expertise. They come to CIRM and say, this is what I'd like to do. Can you vet it for us across the alpha stem cell clinic, the alpha stem cell network? So I think that has been a big plus in the last few years. What's going to happen now in the future? Well, we want to get this technology out across a wider area in the state of California. And that will be the challenge of CIRM. And CIRM will come out with a new program next year. They've indicated that there will be a new alpha stem cell clinic application, and we'll see what that is. But the idea is we want to get to areas that, let's say, are medically underserved. Another way to look at it is we have very sophisticated technologies that exist in non-regenerative medicine areas. Let's say it's orthopedic. But we don't necessarily have the most sophisticated orthopedic surgeons in absolutely every place in California. But people have access to get to them very quickly. And I think that's what we want to do. We want to make sure that the centers of excellence, let's call them, that currently exist, are interacting with other areas in California so that everybody has access to these new technologies. I think the ultimate goal, of course, is the delivery to patients of new treatment, but also the training of the next generation of let's say, clinicians and clinician scientists that will be around for when we are making those trips, let's say, to Mars in the next 20 plus years. So, and I think that CIRM plays a critical role there. Maria referred to the SPARK, of course, program, which brings high school kids really into the arena. But there's other educational systems that are important, but just using let's say these clinics, these alpha stem cell clinics, to generate an area where we can train the next generation will be very important. I'd like to see that happen in the new alpha stem cell program. So I think I'll stop there and let others comment.
Thank you, John. That's really interesting. So talking about building that translational pipeline, one thing we might return to in the Q&A is your sense of how to deal with the technological hurdles of what are emerging as very sophisticated tertiary quaternary kind of delivery systems for some of these therapeutics. You seem to have a sense of how we might reach into disadvantaged communities using that. And that'll be a good thing to explore in a bit. We conclude our panelist part with Mr. Al Rowlett. Mr. Rowlett occupies really a unique perspective in the field in the sense that he's been on the CIRM board. He's also deeply invested in patient advocacy, in reaching out to disadvantaged communities. And so he shares a perspective with us today that comes from lived experience in many different areas. So thank you, Mr. Rowlett. Great to hear your thoughts on this.
Certainly, thank you, and thank you to the other panelists for sharing and providing me with a platform from which to begin. Now, first, let me just say that embedded in the organization is this notion of accountability, and accountability to the unique diversity of constituents and individuals in the state of California. And that's represented in the role of patient advocate. And so I'll describe that very quickly. I'm a clinician by trade, but I've had the unique privilege of working with the Medicaid population. And often the Medicaid population that I'm privileged to serve predominantly has neuropsychiatric disorders. And you heard Maria talk about that being one of the focuses of CIRM's strategic plan going forward, and engaging effectively communities that have been impacted by neuropsychiatric disorders, especially unserved and underserved communities, is in part of the role that I play as a patient advocate. And again, describing it as playing the role is an understatement. Fulfilling the role and doing that work and advancing strategies that have more effectively engaged communities in trials is the value proposition that patient advocates bring to the organization. Understanding the importance of lived experience and appreciating lived experience as you began to look at the social determinants, as were indicated earlier, and the role that the social determinants play in individuals' participation in some of the clinical trials that are sponsored by CIRM that are targeted to address neuropsychiatric disorders, and also the comorbid nature that neuropsychiatric disorders or the symptoms thereof play in addressing other health maladies that we are addressing as an organization. We know this, that we learned quite a bit through the COVID trial, that depression, anxiety, that other disorders impacted individuals in their ability to participate in those trials. And the patient advocate brings a perspective that we help illuminate and provide the applicants of those trials with an understanding of the importance of diversity, equity, and inclusion. And those strategies oftentimes include how to better engage individuals with lived experience. And again, those social determinants require us to take a look at, okay, what role does housing, what role does the implications associated with economics, what role do the implications associated with alcohol and other drugs impact a person's ability to participate in the trials that CIRM is sponsoring? And it is not that we summarily say that, okay, we're not going to include those, but we come up with strategies that better reach out to and engage those communities underserved and unserved in our state, and resulting in them participating in those trials. And also providing for many of the participants in those trials with a unique understanding of the role that neuropsychiatric disorders, how they impact participation. A lot of times, patients with, for example, disorders like schizophrenia or bipolar disorder are impacted by this notion of no hope, no cure. But the understanding that CIRM, through its own work, through supporting research that advances organoid models, might be able to impact and then change the trajectory of a person's experience by way of participating in a trial that brings hope to candidates, that brings hope to constituents. That's part of the role of the patient advocate. And indeed, as you can appreciate from my perspective, there's a lot of passion associated with being a patient advocate. We do so because CIRM's work in advancing trials is, again, what's an inculcated feature is transparency, and making sure that we translate for the constituents in the state of California what it means to participate in a stem cell trial that is impacting neuropsychiatric disorders. Part of my role, my unique role, is as a patient advocate, I ensure that this occurs and is embedded in every proposal that we evaluate, and get to sit with scientists and other clinicians like those represented on the panel and talk to them about the implications associated with communities that are overrepresented in a particular illness, and how we might engage them more effectively so that they might participate in the trial, and we come up with better results that benefit all the constituents in the state of California. With that, I'm going to pause and transition back to other panelists. A lot to say, but let me again conclude by saying this: the work that CIRM is doing in advancing specifically the work associated with neuropsychiatric disorders and alleviating the fears associated with, again, being diagnosed with a disease called schizophrenia and the symptoms thereof is going to be, I think, revolutionary. And then advancing work that includes models that helps better explain that to the constituents in the state of California and considers all those determinants, again, will do a lot to alleviate those fears and concerns that patients have.
Thank you, Ms. Rowlett. That was really interesting and harmonizes well with some of the themes that have come out from the other speakers. As a reminder, please submit your questions through the Q&A function. We currently don't have any, so we have an opportunity to explore some of the things that came out of each panelist's presentation. And Dr. Zaia, I had a question from yours. One of the things we wrestle in my neck of the woods, which is neural repair and stroke and vascular dementia, is the fact that the biology is amazing. The therapies are hard to develop, but as they start to emerge in the pipeline, the delivery systems are complex because they are the brain. And you directly broached that in your talk. It sounded like you had a concept of what some might call a spoken hub model, where you had some of these complex delivery systems in centers of excellence or TQ centers, and you had a spokes and outreach into communities that could feed in. Is that something that you had in mind for how these are actually actualized?
Yes, I think that's a good way to describe it. And there are already examples. For example, hemophilia has its own regional centers. Sickle cell disease has clinics across the state, but they tend to be in focused areas. We have that for many diseases. And I would see, and in fact, the model now with leukemia, let's say you come to City of Hope for your transplant for leukemia, you don't stay here forever. You go back to your hematologist or your general practitioner after the transplant. I see that as one model that gets the let's call it the CIRM supported clinics connected to the rest of the state, let's say, in areas where people need to be served. I mean, the way to look at it is like heart transplants. There are only a few heart transplant centers in the state of California, but really everyone should have access to a heart transplant if that's indicated. So I think the hub and spoke is the best example. We did a transplant, an autologous transplant of genetically modified cells in the last couple of years that was CIRM sponsored. And it was in HIV. So it was persons with HIV/AIDS that signed up for this trial. And they originated from their own local AIDS clinic. They came here for the treatment, but then they went back to their AIDS clinic to be followed for 5 years. Only came here then at 12 months just for a checkup, and then went back. And basically, then it was a trial done in the local community. And so I think that could be reproduced across multiple diseases within the CIRM program.
Yeah, it's really interesting. Kind of with that community penetrance in mind, Mr. Rowlett and Dr. Millan, it seems that one problem a lot of investigators have when they develop clinical trials is essentially the real and sort of damning possibility that they function in a silo, that they develop their community outreach and their trial operations without deep connections to how others have done it and possibly successfully. I'm wondering what you think about the future of CIRM, the topic of our seminar, as a possibility of generating best practices that are universalized and how we might reach into communities, particularly because our challenge is possibly very sophisticated technologies that others outside of California and within the state might use.
Well, again, given Dr. Zaia's example of the heart patient, those strategies that incorporated an understanding of the social determinants of health, what impact does housing, social economics, fears associated with being part of a community that doesn't appreciate or understand the value of pluripotent research and trials and participation, and how to demystify that and engage more effectively, is the role that patient advocacy has, and what the advocacy has in terms of impacting trials that more effectively talk about engaging patients. And just to operationalize that for a moment, there is always a pathway that can be operationalized to get to patients or potential patients in terms of their participation of trials. And it typically includes other individuals with their own lived experience. How that's done is again, in part represented in the role of the patient advocate and being able to talk to and speak to some of the factors that I just articulated.
Follow on to that, as an example of that, the patient advocates on the board actually had led CIRM to develop a way that we consider diversity, equity, and inclusion, and how we fund research and the accountability we have to our scientists, not just accountability, but partnership, really, in enabling our researchers and scientists to think through these elements. So, for instance, in clinical trials, making sure to really test assumptions regarding inclusion-exclusion criteria and the enrollment plans, making sure that you have diverse views on reaching out to communities, because you could have the best science and the best value proposition in the world, but if you are not able to speak to people, they will not participate, and in the future, potentially not accept the treatment. We have seen this struggle even in the COVID crisis, where we now have vaccines with very robust clinical data, yet there is a considerable amount of understanding and trust that's involved in that. So embedding that within how this research is designed, embedding that into how research is deployed is critical. So as a funding agency, CIRM is in a very good position to influence that and create a new paradigm and to shape solutions to these gaps that we find. I have to say that there are actually examples already where even in basic science, this has led to new direction because of this idea of testing these assumptions and not relying on single sources or just common cell lines or HLA backgrounds by which we study disease models. Because why would we do that when it represents when we know in genetic research that the majority of data we have is from Northern European backgrounds, and that is not the world. And so biology is complex, and we need to take that into account from the very beginning. So that is a very real topic that we're already addressing today. It's already incorporated in and evolving and continuing to be developed in partnership with our patient advocates and external stakeholders and scientists, and how science is being evaluated, how we enable by funding creation of new cell lines, by funding the activities that will provide us with a more representative biological understanding, genomics, organoid iPSC models, reference banks, reference repositories, and how you evaluate this in the context of the research. So I think that is one thing about diversity. Regarding access, that was a discussion before. How do you get the new technologies out into the real world, into the communities, it needs to be a true partnership. We need to meet people where they are. There are circumstances where specialized treatments, like organ transplant, where I started my career, there are only certain centers that do organ transplants, liver transplants, pediatric transplants. However, these patients need to be treated by their local physicians. If they have emergencies, they need to be treated by the community care centers. And in addition, they get referred to the tertiary care centers through community care centers. So it's all an integrated, that's medicine, that's our healthcare system. And so the advances that we have seen and will continue to see in the regenerative medicine space means that we now tackle not new issues, but actually bring it into what we have in our healthcare systems, but also identifying what gaps there are and being able to deploy kind of standard approaches. Thank you.
Thank you. Andy, it looked like I have a question teed up for you from your talk, but it looked like you wanted to make a comment there.
Well, I was just going to add that it's the data is male Northern European biased. So we know that organ systems between males and females genetically, in terms of gene expression, are really quite distinct. And there's a whole realm of biology there that's been barely touched upon.
Yeah, great. One question for you, Dr. McMahon. It's very interesting to think, so you mentioned dedifferentiation and reprogramming as some of the interesting biological ideas behind a regenerative medicine concept that doesn't strictly rely on stem cells. And that brings up a lot of the interesting dance the body does between fibrosis and repair in forming true tissue boundaries for exclusion of, say, inflammatory infiltration versus preserving the full integrity and function of an adult cell that's nearby. Wondering what you think about how one moves that forward. In the brain, we can directly change astrocytes into neurons, often with one viral transcription factor. But some of us worry that you will start to alter the balance of tissue boundary formation or scar. And I'm wondering, in other organ systems or from your perspective, what your take is on these things.
So there's some interesting work that was published last year from Scott Lowe's group that used a CAR T cell-like therapy against surface antigens that were present within fibrotic cells within the liver. And basically, the thesis being that you don't want those cells. The best thing to do is eliminate those cells if you possibly can. And the evidence from that paper was that this really improved liver outcome over time. So that's another strategy. You know, you could turn the cells back into something useful, or perhaps getting rid of the cells might, over the long term, be a beneficial route.
Yeah, interesting. We've seen some of that emerge with the concept of removing senescent cells even for aging. That it has some similarity to that.
Tom, there are a couple of questions in the chat.
Yep, thank you. Just about to get to them. The first question here is for Dr. Millan from Dr. Amanda Clark. CIRM has a critical opportunity to be a world leader in creating a permissible framework for stem cell ethics, policy, and regulation in order to garner public trust and participation in stem cell research and translation. What is the plan for a CIRM role in this?
Thank you, Dr. Clark, for that question. That is kind of embedded in the reason that CIRM existed. This was actually in response to the creation of CIRM under Prop 71 was a response to stem cell policy, which initially had it kind of inhibited research with use of embryonic stem cells. And then since then, as you know, there's been continued expansion of the types of programs that CIRM, is funding. And we are going to continue to encounter these types of questions with genetic research as well as stem cells. And we have a standards working group that is a standing working group within CIRM that will be taking up these types of questions. And then from there, recommendations will emanate. We hope to strengthen kind of our collaboration with societies such as the ISSCR and others so that we can address this, very well informed along with the rest of the scientific community. Aside from that, there is nothing specific in terms of a strategic concept that we've rolled out. But we will, continue to be in conversations. I know that a topic has been the idea of where is embryonic stem cell research policy now, and where is CIRM on that. CIRM continues to fund all varieties of stem cell research, embryonic stem cells, induced pluripotent stem cells, gene-modified stem cells, as well as genetic research. And so these will be topics that we will continue to address. Again, the standards working group within CIRM, as well as our board, looking at these questions, give us direction in terms of what we could do internally in terms of bringing programs forward. We welcome any input and that you have as scientific leaders and from the community in terms of any concerns or topics that we should consider.
Okay, great. We're running a little low on time, but we have a really interesting and category-changing question from Scott Pattison, who's basically asking, how are we going to address the manufacturing needs to really scale up and then produce, apply assays for an expected avalanche of cell and gene therapies?
That's an excellent question. It is one of our very important concepts that we're looking at in terms of how we can grow public-private partnership solutions to this, as well as what programs CIRM could fund in order to address what we already know are major hurdles in bringing these amazing technologies from the academic setting and the academic GMP, where the majority of these new technologies are arising, and getting them tech transferred and out into often commercialization pathways. So we actually had an entire manufacturing workshop where we brought in multiple stakeholders. Stay tuned. This is something that's being anticipated in our strategic plan.
Great, thank you. It looks like we've come up to the time limit. This is really interesting. Maybe final word there. I saw a note from Dr. Zaia.
I want to just comment on that. I mean, technology is going to drive the solution to that question. And I just want you all to remember that since December, 268 million people have received gene therapy worldwide. And that's because technology changed in the last year. And we now know how to deliver genetic material in a nanoparticle.
Great. Thank you, panelists and audience, for great questions and engagement.